I'm going to very, very briefly go through this, but really like what I want to do with everyone is a session on using the lab exchange. So I'll get to actually what that is. And hopefully you guys will all be able to participate if you're interested. So it's kind of try to make this a bit more interactive. I think I can get this done earlier than 40 minutes. So hopefully that helps with getting things a little bit more on track. So I will try to do that. Briefly, what is LabDAO? So at LabDAO, we provide and use open tools and infrastructure for computational life science research. It's extremely broad, and that's intentionally broad. We are figuring out how a scientist could work remotely from anywhere, collaborate with anyone from anywhere, and work in a decentralized manner. In the future, that looks like going from ideation to completion in terms of an experiment. But to start off, we have to start somewhere, and we started by focusing on computational biology tooling, which, if you're not familiar, that is pretty much all online, or sorry, on your computer. So there's less wet lab, which is things where, you're actually doing the things in the lab, and more computational sides of it. It's pretty self-explanatory. So this allows us to work explicitly with tooling. So that would be, for example, a tool which takes a protein, does something to it, and gives you an information, something on the other side. So think of this as like an application or something, if that's, that's a very reductionist way of thinking of it, but it's something that allows you to do something to biological data and get some information from it. And if you aren't familiar with computational biology, I definitely encourage you to do some research on it, because that's a very interesting area right now, and there have been some really cool advances with AI lately, which we can touch on actually as well if people are interested. Big questions normally is like, how is this DeSci? So pretty broadly, we are using IPFS for storage. Sorry, I'm just getting over a cold. We're using IPFS for storage. We're using, so that's interplanetary file storage. So it's pinning it, distributed storage. Again, if you're not familiar with that, definitely encourage you to look into it, but I don't have a lot of time today, so I'm not going to go super deep into that. Backlog is a really cool project that actually allows people to have their pin data on IPFS and actually do the compute where the data is stored. That's really helpful for scientists, for example, who have massive data sets that don't want to be downloading them every time they have to do compute. So it really helps streamline and make everything faster. So that's also like a Web3 tooling. And then in the future, we're going into like permissions, ownerships, everything that is generated on the lab exchange, which is where we allow computational biologists, or we enable computational biologists to run their tooling. Everything will produce an NFT. It all has these content addressable identification. So it's really traceable, really great for proof of ownership or who worked on what. There's, I don't know if anyone was at Paul's talk earlier, but there's really cool IP implications there as well. So there's a lot of really exciting ideas that are coming out of this. So today I'm going to talk about the lab exchange. So this is up until recently, this was a command line interface. So, a lot of computational biology is run just on the command line. You're writing your code, etc. But we have recently moved on to building a very, very MVP front end. So really exciting. You guys are going to be some of the first people to ever try this out. Maybe something will go wrong. We'll see. But we want to be able to show you guys a little bit of what we've been working on. So with that, I'm going to leave this up. Wait, just a second. I'm sharing my screen still, right? Any comments? Is this working? Yeah. Can I get a yes from anyone here to let me know that everything's, you can hear me at least? All right. I'm going to have to trust that you can hear me. Amazing. Thank you. Okay. I know there have been some technical difficulties, so I just want to confirm that. All right. So I am going to get, actually, I'm going to ask the moderator if you can assign Akash to this stage because he's having an issue. He's not assigned to a stage yet. So if it's possible to get him in here. I don't know. I'm sorry. This is probably really bad for everyone. Let's go back to this. So I don't know if it's easier. I'm going to share this link in the chat for this. Oh, I can't. That's a search function. Private chats. Okay, I can't do that. I'm going to have to ask everyone to type this in. So here we go. It's not that long. You can type it. I'm not sure what the best way for me to share this is. I can share it in the YouTube comments. Okay, I think. There we go. Cool. And damn, that's very confusing. All right, I'll give everyone a minute to open this up. Let me check if. All right, while we wait, I will also open up. Cool when you can link things like that, but that was the wrong one. prod.labdow.xyz. All right, let's go. So I'll log out. Everyone should be seeing this screen. We are using a, oh, Akash is here. Hey, this is Akash. I don't think everyone can see him, so I'm just gonna share my infinite screen just to say hi, but he can join in the commentary. Can someone in the YouTube tell me, Akash, can you say something? Can someone on the YouTube tell me if you can hear Akash, please? Okay, well, we'll see. Potentially. I think it's there's a little bit of lag on the YouTube, so we'll find that out in a minute. Okay, so we are on app.prod.labdow.xyz for anyone that hasn't gotten the link yet. And we're going to log in. So we are using Privy. I think, Akash, if you want to say anything about that for login. This is a cool Web3 feature here. I'm going to get you to click on tasks. And today we're going to be doing some protein design. So if you click on this, we have the GitHub for this tool and the paper for this tool here. I'm not one to speak to the complexities here, but you can do a ton of research on your own if you'd like about how this tool is working. But it is using RF diffusion, which I think some of you may have heard about if you're following like the AI biospace. And essentially, like, we are doing some protein design. So Akash also made a really great analogy yesterday about how this works, if you want to share that.
Success of any therapeutic R&D, being able to generate novel binders, novel proteins, have some validation prior to going into the wet lab and eventually, animal studies, clinical trials. This almost de novo approach is absolutely critical for, I guess, the future of developing medicine. And, I think it sort of it plays really well with the open nature of Web3 and DeSci as a whole. And so we're trying to make these tools available to as many researchers as possible.
Awesome. So with that, again, I know I'm just being vague and saying you guys should research this, but I do really encourage you to look into this because it's a very cool space and there's so much information to learn, even as someone who doesn't have too much biology background, you can just get an idea, or if you are like a computational person, you can get an idea of like how exciting and innovative this space is becoming. So with that, I'm going to encourage everyone to navigate to this page. So again, if you weren't here before, you click on tasks, protein design, and you'll be brought here. So the name of the experiment, you can name it whatever you want. I'm going to call mine Bankless Unconference test. But yeah, name it as you will. We're going to leave the model as is. You can tell that we have a lot of models right now, but these are just like slight variations of the same ones. Again, this is a very like, you guys are some of the first ones to try out this tool. So there's a lot of changes that we're going to be implementing, but the number one thing that we need to change or add right now is a protein. So PDC UPAR1 is the protein that we're going to be using. So that is already added. So it's already pinned to IPFS. It's in this space. This is all publicly available. For example, like this tool, if you wanted to add a tool to the Lab Exchange right now, you would be able to. You write a manifest for it, and you can see, like, for example, this is a tool manifest for this one. It's just like in a JSON format. Here we go. This is what this tool looks like. It's in the right format and it's in a container. So it was able to be added to the Lab Exchange. And now it's here and it's like automatically usable by anyone. These proteins, for example, the protein that we're using today was already added by someone, but conceptually we could be using a protein that was generated by someone doing an experiment, or Akash could add a protein. I mean, this is exactly what we're doing. Akash added a protein and it's on the Lab Exchange now. So I'm able to use this protein in any experiment that I want.
Yeah,
So
and
I'm going.
quickly
Oh,
add
go
to
ahead.
that, Lily. One of the one of the really powerful advantages that Web3 and decentralized tech such as IPFS enables is it enables like natural default collaboration. So if you think about like traditional academic systems or even traditional companies, it's not so easy to share your results and share your data. By leveraging content addressed data, which exists and is fetchable by anyone that has an internet connection, you really create this almost like biological soup where, you know, if I design a protein, Lily or anybody in the audience can run small molecule docking experiments on it, or even mutate it to try to create a different flavor of that protein. And if you just like expand this, and a lot of people are building on this type of system, you really get an absolute explosion in the space of innovation that is possible. And I think this is, you know, I think this is probably going to be the future of how people do this early stages of R&D. Um, yeah, content
Totally.
addressing is really amazing.
Yeah, I mean, we could talk for hours about content addressing too. So if anyone is interested in that stuff, definitely hit us up as well. Because I agree, I think it's one of like the coolest things that we have going right now. But yeah, so if you can click on here, search PDC underscore UPAR underscore 1. So that's the protein that we're going to be using today. Okay. The target protein to design against. We're going to keep this as A. So this is the target chain that we're going to design against. Keep this as 1. This is the start residue of the target protein. I would love to go into what these mean, but it would take probably a couple of classes. So for now, I'll just tell you these are numbers that you can modify. I don't know if anyone is familiar with Hugging Face, but if you've ever used or like Replicate, like these tooling where you can do some AI simulation, and there are sort of boundaries that you can set. Similar energy here. It's not exactly a one-to-one, but you can conceptualize it similarly. So for the end residue today, I think we were going with, we're going with 200. So input 200 here. Hotspot, we're just going to leave that as is. It's an optional field. And then, so this, now we're talking about the protein binder that we're designing. So this is like the AI design protein binder. We're going to go with 40 on this. And then a cool feature that we have is any of these, if I wanted to add another one, I could. And then we have a combinatoric sort of experimental run. So you have multiple experiments that are running simultaneously. So all of these things are happening with this. And then we try it against this, which is a different number. Let's say like 3 or something. And then I could add another one. And I could add another one. And I could add another one. And as you can see, like the number of experimental runs goes up and up and up. So we have a number of different experiments that we can run. For now, I'm just going to add a second binder length. So I think last time we tried was, 80. Let's go with 80. So just click that little plus button there, type in 80, and you can see that the number of experimental runs, you can see that we actually have 2 different types of binder lengths that we're going to try out, see how that goes. And we're going to leave the number of binders at 2. Leave these advanced settings. You can do some pretty cool stuff with this too if you have like the knowledge of how these tooling work. But yeah, we're definitely going to leave that for now. So, all right. At this point, I'm going to ask if there's any questions, and if not, we're going to submit it. Questions? I don't see any here. Let me go to the. All right. Oh, okay. Wait. Much better than collaborative. Oh, thank you. Interesting tool fit. Awesome. Great to hear. So I'm minimizing this guy. Sorry, my windows are a little bit overwhelming. We're back here, and we're going to hit submit to experimental runs. So I'm clicking that. And then we're immediately brought to the page where this experiment is running. So you can see that the status is new here. This is the model that we used for it. And everything's given a job ID. So that is a backlog job ID. We can go into what that means too if you want. But for now, just know that every job in your experiment has a unique ID, as well as a CID. So this is the CID. This is a content adjusted content identifier for the experiment that you just executed. And this is the wallet address that I have. So if anyone wasn't here for the beginning of this, Akash briefly went over how we're using Privy, which is cool because it will generate a wallet for you if you don't have one yet. Or you could use your personal wallet as well to log in. So I had it generate one based on my email, and this is my wallet address now. And that's all of the work that I do on the Lab Exchange is linked to this wallet address. Sorry. So I'm going to hit update in this top corner. We can see it's just running now. This is the name that I gave my little experiment. Your experiment we can go into one of these jobs actually. We have 2, right? Because remember we used 2. We used 40 and 80 for the binder length. So therefore there are 2 jobs. We're trying 2 different experiments with this. But I mean, you can imagine one where you like the combinatorics are really large. And you can get up to like, I don't know, however many runs that you could imagine basically. So that could be a very long list. But yeah, so we can see like the experimental parameters that we inputted here. This is in a JSON format. And then we can explore the tool. Currently no errors. Let's update. In the top right corner, I'm clicking update. All right, it's still running. All right. No results yet. This is where our outputs will be. But
must have a lot of people submitting jobs because it looks like our compute nodes are still picking it up. I mean, one of the interesting challenges that I think DeSci will have to overcome is the scalability piece. But I think for anybody that has submitted an experiment here, just keep this tab open and just periodically press update. The compute will run on the compute nodes. I think once the queue is cleared out right now. Looks like it's a little bit clogged up at the moment.
But we do have like there's no errors yet, so that's very good. It means that the experiment that we did is probably chugging along. We'll probably be okay. We'll see. But for now, what we can do is we can download the input that we use. So that is that PDCU .pdb. A .pdb file is a protein file, generally speaking. So I'm just going to click on that. Oops. And there we go. It downloaded for me. So we could do a few things while we wait. First, I'm going to ask if there's any questions. Do we have any questions on here? Oh, here we go. Sorry. I totally missed these. Oh yeah, visualization. Cool. Yeah, definitely. Awesome. So here we go. We're here. We downloaded this guy. So I just clicked on it, opened a new tab, and it downloaded for me. If you guys, again, to show you where we are, we went from the experiment that we just did, and we clicked on one of the job IDs. And then I looked at my input, and I downloaded it just by clicking on it. While we wait, I'm going to ask everyone to go to molestar.org. So that is molstar.org. Let's go there initially. And then we can open Molestar Viewer. In the near future, we'll actually have a little integration in this app for this, which is kind of exciting. And I'm going to go, oh, look, this is my downloaded guy. So this is the input that I gave in. And I'm going to pull it directly into the window here. It's just loop like that. And there we go. This is the protein that we're using today. You can see it. Very cute little orange and green guy. Simple. Some little curly bits here. You can play with this. I don't know. Poke around a little bit. This viewer has some cool features. Once we have the output, we're going to put it in here as well. And we'll be able to see how close it comes to the protein. Sorry, I'm like trying to reduce this into simplicity. How it interacts with the protein, let's say that. So let's check back on this. How we're doing, Akash? Any updates?
I think the queue is still running. However, I can send you also a link of a completed job that could be useful too.
So I do have a completed job here. So this one is completed. This is what it will look like when it's complete. It's going to take a few minutes. I mean, we're doing some pretty intense computation, and we're all doing it at once. Sorry. But here we go. This is a completed one. We did this, I think it was like yesterday or something. Once it starts running, once the queue is like cleared up a bit, we'll be able to like actually follow along with the logs, which is quite helpful if you're used to just seeing like a command line interface in order to interact with your tools and the protein outputs, etc. So if you know what this means, it is helpful. Otherwise, we can just scroll past it. And here for this one, we can see that we had this input. I'm just going to download that input. And we are given a bunch of outputs here. So these are various designs that were outputted by the experiment that we executed. A lot of them, actually. What I'm looking for right now, and this is kind of like a standard thing in computational biology, is default best PDB. So best is kind of going to be the one where it's like, this was the best one. I can go to detail. Well, actually, I can't go to detail about why that is, but it's
various metrics for binding that the container uses, BanklessDeSci uses. This is like the generative best binder that was created based off of the protein that we were given. So I think, Lily, if you download that default best one,
yep,
you can throw that into Molestar.
Yeah, we're going to open up the viewer. So here we're going to do the same thing as what we just did. I'm going to take, this is my input. So slightly different looking protein, but same idea conceptually. This is the protein I put in. And then here's my output default best. I'm going to put this into Molestar Viewer as well. And look at that. This is what was designed to interact with my protein. Very interesting. this was given a number of different parameters. This is like what was generated using these tooling. So for if you're not a computational biologist, you might just be looking at this and being like, cool, squiggles. um, which is fair. You do need pretty much at least an undergrad, and definitely a master's would be helpful for this to actually get any useful information out of this. But what I can tell you is it's really cool that we were able to do this using decentralized compute, decentralized storage, completely open protocol, and anyone can replicate these experiments as well. So let's check back just one more time to see. I want to like ask if anyone is able to like how the queue is going, because I know I'm pretty far behind in it. But I want to see if there's any questions. Oh, there's a YouTube channel question. Interesting. Okay. Yeah, that's I'm gonna stop sharing my screen for a minute. This is me. Hello. It's a really big picture of my face. Yeah, I think that really like the exciting part of this for someone who isn't a computational biologist is knowing that this tooling that is first of all like oftentimes very hard for scientists to share between each other, even because even just like getting it set up and installed is quite complex. Files that would have to be shared in various methods, collaborations that would maybe not happen, are all going to be quite easily enabled by this tool that is just starting to materialize into something quite usable. So I'm quite excited about that. I think, Akash, is there anything else you want to share?
Um, Nothing that comes to mind right now. I guess this is the first time that we were like publicly showcasing this. It's a sneak preview. We'll definitely have like a larger, more fanfare
Yeah.
announcement for this application. But for those of you who are lucky to see it, feel free to hop in. It's once again it's app.prod.labda.xyz, and yeah, if you end up designing any like protein binders, uh, let us know. Tweet at us what you've come up with, because I think this is going to be the future again of how R&D works.
I totally agree. And if you do have any feedback or any questions, or just want to chat in general about this stuff, feel free to send me an email. I'm also on Telegram at LilyHG. So that's the same as my username right now. Actually, I'll type that out as well. Here we go. LilyHG on Telegram as well. Yeah, reach out. We're really excited about this, and really excited about how in the future it's going to continue to grow. We have some really big plans for it. So yeah, and if you have some computational biology tooling that you are interested in either putting on the Lab Exchange, or you think that it should be there, I'd also love to hear your feedback on that as well. My Twitter handle is the same as my Telegram as well. Yeah. And I guess I'll just say, like, any questions, like, open that up one more time. And if not, I'll pass it on to Valeria. Sorry. Boop. Oh, yeah. Yeah, the Explain Me Like I'm Five. We're working on that. That's Elijah says, you'll have to give me the Explain Me Like I'm Five. I think that's one of the hardest parts of science communication for sure. Is reducing it to the basics for a 20-minute to 30-minute talk.